Welcome to the Antibodies Lab. Today we're going to explore one of the most diverse and immunologically important classes of molecule that none of us can live without. We'll discover how antibodies are involved in human health and some diagnostic techniques we can use to help patients. A young couple are visiting the office of one of our collaborating physicians, Dr. Lee. They have some questions about Rhesus disease. Dr. Lee has asked if you'll attend their appointment, so let's head over to them now.
Hello, I'm Dr. Lee, your consulting obstetrician. How can I help you today?
Hi Dr. Lee. We are here because we are pregnant with our second child and are a bit worried.
We've read about Rhesus incompatibility and that this could cause problems during my pregnancy.
Can you tell us more about Rhesus incompatibility and the risk for our baby?
Do you know when Rhesus incompatibility can cause problems? Exactly. When a Rhesus-negative mother is pregnant with and gives birth to her first child, she may be exposed to Rhesus-positive fetal blood. The mother's immune system recognizes the Rhesus antigens on the fetal red blood cells as foreign and begins to produce antibodies against them. This can cause immune issues during a subsequent pregnancy. When a Rhesus-negative mother is pregnant with her second child, anti-Rhesus factor antibodies raised as a result of a previous pregnancy can cross the placenta and trigger an immune reaction in the growing fetus. Maternal antibodies enter the fetal bloodstream and bind to red blood cell Rhesus antigen if expressed. Bound cells will be destroyed, resulting in hemolytic disease of the newborn, or HDN for short. Why is the red blood cell count typically decreased in HDN? That's perfectly right. This process is a type of hemolysis. Red blood cells carry oxygen to all parts of the body, so low red blood cell count would mean the growing fetal tissues and organs may not get enough oxygen, which can be fatal.
That sounds really dangerous. Can we test to see if our baby will be affected by Rhesus incompatibility?
Absolutely. I will schedule blood tests to check your Rhesus status to see if further treatment is needed.
Using the lab pad is an important way to get the most out of this simulation. Is this the first time you've played a labster simulation? Perfect, then let's get started with your first mission. So, Joel and Carmen are expecting their second child and want to find out if there is Rhesus incompatibility between Carmen and the baby. Dr. Lee has tasked you to review their case, so let's get started. Click on the lab coat rack to wear a lab coat. Awesome. Now that you're safely dressed for the lab, let's help Joel and Carmen.
Hey there, I'm Marie. Before we get started, let's talk about the key players in our mission, antibodies. Antibodies are super important when it comes to protecting us from viruses, bacteria and other pathogens. Antibodies recognize specific molecule features called antigens. Once bound, they form a unique antibody-antigen complex. It may help to imagine the antigen as a lock and the antibody as the specific key that fits it. If antibodies and antigens can bind together, which statement is true of antigens? Well done. An antigen is any molecule that can cause an immune reaction. These molecules range from proteins, nucleic acids to whole organisms. Have you ever had hay fever? Then the antigen was probably pollen, which stimulated your immune system and made you sneeze. Antibodies are also known as immunoglobulins, or, for short, Ig. They are Y-shaped proteins with hypervariable regions that recognize specific molecules. Let's have a look at antibody structure in the molecular viewer.
Go to Workbench 1. Click on the Antibody button to observe the structure of an antibody. Here you can see a hologram of an antibody, just like the one which causes problems in Rhesus incompatibility. Isn't it a beautiful Y-shaped molecule? An antibody consists of two long chains called heavy chains and two short chains called light chains. Can you see the different chains in the hologram? The two heavy chains wind up the molecule from the bottom. One fades from green to turquoise in color and the other fades from red to orange. The two light chains at the top of the molecule are colored yellow and blue.
How many polypeptide chains build up an antibody? Right on. You're a great observer. There are two identical heavy chains and two identical light chains. The chains are connected by disulfide bonds near the center of the molecule. Every antibody is unique, like a fingerprint. They are produced to fit and detect only a single antigen. Binding of the antibody to the antigen happens at distinct regions at the tips of the Y, also known as the variable regions. These regions are different for every antibody and specifically bind to what's called the epitope of the antigen. In mammals there are 5 different classes, or isotypes, of antibodies: IgA, IgD, IgE, IgG, and IgM. They are categorized according to their biochemical properties and fulfill different functions in the body. See how the classes differ in their shape and organization. According to the theory, which antibody isotype crosses the placenta? Genius, IgG is the only antibody in the plasma that is transported through the placental barrier. Other monomeric antibodies, such as IgD and IgE, are typically not found in blood serum or are present at only very low concentration. IgG with a variable region that specifically fits the Rhesus antigen, also known as D antigen, can bind to red blood cells when it is expressed. Remember this well. It will be important later on in the blood typing test. Let's see if you can identify the structures of an antigen and an antibody.
Click back to return to the lab center and head over to Workbench 2. Click on the laptop. Label the different parts of the antibody and the antigen. Use the theory for help if you need it. Perfect. You have labeled all the parts of the antibody and antigen correctly. When an antibody binds to its matching antigen, it forms a tight complex. There are 2 parameters which determine how efficiently an antibody can bind antigens. They are affinity and avidity. Affinity describes the strength of a single antibody-antigen contact site. Avidity describes the sum of affinities of multiple antibody-antigen contact sites. IgG has a strong affinity for its antigens. IgM, on the other hand, has low affinity but high avidity due to having 10 binding sites compared to IgG's 2. How much higher is the avidity of IgM compared to IgG, assuming that both isotypes have the same affinity? Fantastic! IgM has 10 binding sites while IgG has only 2 binding sites. Let's watch an animation to understand how the mother's immune system specifically produces antibodies against the Rhesus antigen of the fetus and how an antibody-antigen complex is formed. This is a fetal red blood cell. On its surface are Rhesus antigens. When Rhesus-positive fetal blood is first encountered, the Rhesus-negative mother's immune B cells raise antibodies to the epitope. Initially produced IgM can't cross the placenta, but IgG is produced later. The mother's immunological memory is not set. During her second pregnancy, anti-Rhesus IgG can cross the placenta, recognize fetal antigens if expressed and trigger an immune response. IgG will form a tight bond with the D antigen. Let's take a closer look at what happens at the antibody-antigen complex interface. In every antibody-antigen complex, there are 4 main types of bonds that can be formed. Hydrogen bonds, electrostatic forces, or ionic bonds, van der Waals forces, hydrophobic interactions. Which cell type produces antibodies? Marvelous! Different antibody isotypes are produced at different times. IgM is typically produced first during an acute response. IgG is produced later by cells that maintain an immunological memory. The fact that IgM doesn't easily cross the placenta is why a first pregnancy typically isn't affected in the same way as the second, where rhesus incompatibility exists. What are the 4 types of bonds in an antibody-antigen complex? Genius. Bonds formed between an antigen binding site and an epitope are weak, non-covalent bonds. In contrast, covalent or peptide bonds are very strong bonds that require high energy to break. Awesome. You now understand the core concepts of antigens and antibodies. Nice work. Before we determine the rhesus status for Carmen and her children, we need to figure out how to process the blood test and interpret its results.
The blood typing test we will use is based on the 2 most commonly used blood grouping systems, the ABO group and the Rhesus group. Depending on the presence or absence of A and B antigens on the red blood cell's surface, a person either belongs to the A, B, AB or O group. People testing positive for D antigen are assigned as Rhesus positive. Following this classification, a person with blood group A+ would have A antigens as well as D antigens found on their red blood cells. Do you know your blood type? Knowing a person's blood type is extremely important in the case of blood transfusions. When a patient receives blood from a donor with an incompatible blood type, agglutination will occur. This means that the red blood cells will clump, which could be lethal to the patient. For example, if a person with blood group B donates blood to a person with blood group A, agglutination will occur in their blood vessels. This happens because the recipient will produce anti-B antibodies to the previously unencountered antigen on the donor's red blood cells.
Head over to Workbench 2. Let's get started with the blood typing test and determine the blood type of 4 different patients who are waiting for a blood transfusion from a matching donor. There are 4 Eldon cards in front of you with circles on. Each circle is pre-treated with antibodies for a specific blood antigen. If a matching antibody-antigen complex is formed when blood is applied, we'll see agglutination. From there, we can determine which blood group the sample belongs to. Pick up and wear some gloves to protect your hands in the experiment. Pipette a drop of water onto the circles of Eldon card 1. What is present in the test circles on the Eldon cards? Precisely. Anti indicates an antibody. The last circle is left blank on purpose, and it's called a control. What do the antibodies in the anti-D test circle detect? Awesome. This circle will later help us to determine if the rhesus status is positive or negative, that is if the rhesus antigen, or D antigen, is present or absent. Pipette blood sample 1 onto the circles of Eldon card 1. Use an Eldon stick to mix the blood sample within the test circles of card 1. What is the significance of having a control? Marvelous. Experimental controls help us to check if our kit is working correctly and to exclude the presence of potential contaminations. Wait for 10 minutes. Click on the arrow to observe the result of the neighboring card. Discard the mixing stick and your gloves. Well done. Look at the test circles. Some of the blood drops don't look as homogeneous as before as the red cells have clumped together. This is called agglutination. What happened when the blood agglutinated? Exactly. Binding of the antibodies to their corresponding antigen on red blood cells causes multiple cells to join and form large complexes that will appear as blood clumps. Agglutination simply means the clumping together of particles. In the context of blood and serology, agglutination is when antibodies bind up antigen on the surface of different blood cells, creating a blob-like mass of antibodies and cross-linked cells. This is different from blood clotting, which results from a tightly regulated response involving many signaling molecules. Why can agglutination be lethal for the patient when it occurs during a blood transfusion? Well done. Blockages in blood vessels could shut off the blood supply to organs and tissues, a potentially fatal outcome. Okay, let's try identifying the blood types on the Eldon cards you processed. Label the blood types on the cards. Click and raise your lab pad when you're done. Have you labeled the test cards? Excellent. You're clearly an expert on blood typing. You now understand the principles of compatibility in blood transfusions. We just received blood from a donor with a B-negative blood type. Could any of the patients we've just typed receive blood from this donor? Bravo! Blood from a donor with blood type B-negative would cause agglutination in patient 1 and patient 3. This happens because patient 1 and 3 produce anti-B antibodies that will agglutinate the donor's blood. Why is blood type O-negative called the universal donor? You got it. Blood type O-negative blood cells do not have any cell surface antigens. The lack of A, B and Rhesus antigens in an O-negative donor's blood means no antibodies will be raised to the blood cells during or after a transfusion. Let's apply these principles to help Joel and Carmen. This way, we can determine if there is Rhesus incompatibility between Carmen and her second child. Click back to return to the lab center. Go to Workbench 3. To determine a potential Rhesus incompatibility, we first need to identify the Rhesus status. Let's recall. What does HDN mean? Brilliant. Remember that HDN is one of the problems caused by Rhesus incompatibility. Hemolytic can be broken down into two words, heme, meaning blood, and lytic, from lysis, meaning to split or break down. We have collected the blood sample from Carmen and her first child. We have also sampled the fetus by withdrawing blood from the umbilical vein. Let's perform blood typing for Carmen, her first child, and her unborn second child. The lab assistant has already prepared the Eldon cards with a drop of water on each circle. Pick up and wear some gloves to protect your hands in the experiment. Pipette a drop of blood onto the test circles of Eldon card labeled Carmen. Use an Eldon stick to mix the blood sample within the anti-test circle. The lab assistant will prepare the rest of the blood samples for the remaining Eldon cards. Observe the cards when she is done. Wait for 10 minutes. Click the arrows to observe Eldon cards for Carmen, first child, and fetus. Discard the Eldon stick and your gloves. Label the blood types on the cards. Click and raise your lab pad when you're done. Have you labeled the test cards? Well done. Now we know the Rhesus status for Carmen, her first child, and her unborn second child. Can you determine if there is a Rhesus incompatibility between Carmen and her second child? Good job. We know that Carmen is Rhesus negative, her first child is Rhesus positive, and the fetus is Rhesus positive. This could potentially be harmful to the fetus if there is no further action taken to treat the Rhesus incompatibility. Have you noticed that Carmen and her first child don't have compatible ABO blood groups? Why does an ABO incompatibility between a mother and her child rarely causes severe problems? Nice. In contrast to Rhesus antigen, IgG isn't usually raised to A and B antigens. The IgM which is sometimes raised can't cross the placental barrier. In rare cases, and especially in mothers with blood group O, IgG anti-A or IgG anti-B antibodies are naturally present, which can cause mild and short-lived symptoms in newborns.
Let's review what we've learned. We know the basic concepts about antibodies and antigens, and that compatibility is important during blood transfusion. We also know that Rhesus incompatibility can be a problem during pregnancy. We can now confirm that there is Rhesus incompatibility between Carmen and her child. Let's go back to the hospital consultation room to tell Joel and Carmen what we've found out. Hi, welcome back. We have determined the Rhesus status for you and your children. Carmen, after performing blood tests, we have found that you are Rhesus negative, while both your first child and unborn second child are Rhesus positive. This means there is a Rhesus incompatibility.
Oh dear, is there any treatment?
Yes, ROGAM injections have been developed to treat Rhesus incompatibilities during pregnancies. These injections contain low doses of anti-Rhesus antibodies, which do not harm the fetus, but prevent the maternal immune system from producing additional antibodies. Ideally, these injections are administered during every pregnancy, but further tests suggest you are not yet Rhesus sensitized, and so this is still a viable treatment option.
Sounds great. We would like to make a schedule for the injections. Thank you so much for your help.
Congratulations. You have completed the antibodies lab and helped Joel and Carmen determine the Rhesus incompatibility. Carmen has given birth to a healthy baby girl. Let me ask you one last question. Do you feel like you gained relevant knowledge during this simulation? Thank you for your feedback. Your score has been saved together with your 100% progress. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.